Okay, genetics and inheritance, guys. Welcome to my class. My name is Mr. Mpepu, and today we'll be doing genetic engineering. As you can see, the date there, today's date is the 8th April 2024, Monday, and the time there is 12:38. So today marks the beginning of week 4 and the end of genetic and inheritance chapters, right? So with that being said, sanbonani, dumelani, afsheni, hello, nda. So guys, welcome to my class, welcome to my class, welcome to my class. And then in this class, I was very much excited yesterday after finding out that we are 43 students in this class. So it means that our class is very much fully packed. I started this, guys, at the 18 week 1. If you still remember, it was the 18th, and then up until today, guys, beginning of week 4, read Tomaka the 8. If you can go to the calendar, or if you can go back to my YouTube videos, you can see the date that these videos were uploaded. We were very much strict and going with our plan, Monday up until Friday. So guys, thank you for thank you guys, those who are joining us for the first time this side. We welcome you guys. The space are still there. We still have space. We can still add more decks, or we can right? We can move, or we can shift for you guys, or we can have this entire class. You can have all this class here full or packed of you guys, right? Another thing, let me take this moment, guys, to shout out to this person here, very affectionate, chocolate. They said affectionate chocolate bon bonds. I'm not sure what's this, right? So this is the email that, guys, this person commented on my last video saying that week 3 we are done. So guys, whoever you are, I want to appreciate you guys. I want to appreciate you. And then those 43 of you who subscribed, guys, I also would like to appreciate you. Then we can have our class here, our imaginary class, and then we have these decks, guys. We still have space. You can still come. We can still register. We are still registering new students, guys. We are only 43 plus me, then we'll be 44. Then today, let's add another students, guys. Subscribe, guys. Those of you who come to this school via the TikTok street, we welcome you. And those who reach this class via the Facebook street, we welcome you. And then also those who reach this class from just the word of mouth. Maybe someone in this class told one of the students outside of this class to say, hey, we have Mr. Mpepu there, and we are learning life science in a different way, or in a, do you understand, in a way that you can sit down, chill, have your drinks. Do you understand? Put the earphones on, reduce the volume, or whatever you want to do, and then listen to this life science, right? So you come here, we welcome you guys. The space is still on. We are still registering. Come and register to vote. Come and register to be counted as this class, guys. 43. We are only 43. So guys, another thing, don't forget to comment, guys. Don't forget to comment. Don't forget to like. Don't forget to like, guys. Don't forget to subscribe there. And then another thing, one last thing, don't forget to tell others, guys. The word of mouth can also work in this situation, right? Let's add other students, guys. This just show the potential of this class, or this class can do what, can increase, right? We started together, guys. Now I'm studying this thing. It's not like I know this thing by head. I'm studying this thing. By the end of December, I'm still promising you 43 students that I'll be sitting down writing exam year 2024. So that will be my promise. I'll make sure, I'll apply, I'll make things, I'll go and talk with other people that make things to be possible, and then I sit down and write this exam with you guys, right? Do you agree? Not a rewrite. If I fail, I fail. But if I pass, I pass. So it means that if I got 40%, I believe that, I know that all my students, there's a possibility of them to get this 40% out of this paper, because we did these things together, right? So thank you for that, guys. I just wanted to to measure that 5 minutes there. Okay, back to the class, back to our class. Thank you those who are subscribing and those who are commenting, those who are liking. I say thumb up, guys. Do this also so that our videos will be what do we call, will be suggested to others, right? So that others will also join, and like that. So thank you, guys. That being said, week 2, week 3, okay, thank you. Week 1 we did the concept of inheritance, and then monohybrid crosses, sex determination, sex linked, and then week 2 we did diabrid crosses, and then blood grouping, and then week 3 we did genetics and lineages and mutation. So in this mutation, this knowledge that we learn here in this mutation is now pushing us to come and learn the genetic engineering. What is this genetic engineering? The paternity testing, you did it when you were doing protein, DNA synthesis and what, right? DNA replication, where you learn that we can use this DNA to trace the relativity between a daughter and the father, or between the offspring and the father, right? And we can also use the blood grouping to identify or to exclude someone as a father of an offspring, if you still remember here, right? So you learn these things. You learn that we have a chromosome, and in that chromosome we have an allele. In that allele we have a gene, and then when we say a gene, we're talking about something like this double helix structure, right? Where this is going to be unzipped, and then this will have to produce a string of a DNA called mRNA that will go and attach to the ribosome. After attaching to the ribosome, we are going to produce a certain protein, right? So here you were learning how this gene is being passed from one parent to the next, right? Here we learn the concept, the heterozygous, homozygous, a phenotype, genotype, right? So after that we learn the diabrid crosses. What if we have two characteristics, right? The blood grouping, how do we group these individuals together, right? This is what we were learning all along. How do we understand if there's a gene of a blood, how does it differ with another individual, right? And then from there we came and learned the pedigree. What is a pedigree? How can we trace these genes that are being passed from one parent to the next parent, right? We used the pedigree to study those characteristics being passed from one generation to the next. You'll agree with me, right? Then after that, we learn about mutation. We learn that no, man, is this chromosome of ours, and then we can say that we have an allele, we can have a gene, but there's a problem when this gene of ours, it get changed, or it undergo a random changes, right? So it means that we have a gene mutation, or we have a chromosomal mutation, where a structure of a chromosome can be altered with, or can change, or the number of these chromosomes can change, right? Trisomy and monosomy. And then we also have a gene mutation, that in this gene mutation, some nucleotides can be added, or some nucleotides can be removed from a DNA structure, right? And then we are dealing with a mutation, or a change that is happening on a genetic level, right? So with that information and mutation, oh man, naturally, or with some x-rays, or with some chemicals, we now see that this DNA of ours, it can be removed, or we can add something here. How about we come and utilize this information, or we utilize this knowledge of adding and subtracting of genes here, and we use it to benefit ourselves, right? For an example, we go to the chickens, and then see this example here, I will be using more chickens and beef, right? We have chickens that 3 days, 4 days, 5 weeks, they are now matured and ready to be eaten, right? So you ask yourself, so it means, no, we conclude, or we know that, or we can conclude that there's something that they're giving those chickens, so that those chickens can increase in a size 2 times than their normal breeding time, right? So that is a genetic engineering. That is the engineering of genetics, right? So we can move from our ATP. Now we understand what is the genetic engineering coming, and why is the last topic here under this chapter, right? So from here we can move to our KEBS document. Our KEBS document, there's nothing much here. They said we have to do mutation. After learning mutation, after understanding mutation, that this mutation is changing a gene level, or it's changing a chromosome, and with this change, it means that we can ourselves now come and manipulate this gene. When we say we manipulate gene, it means that we can come to your gene and cut using a certain enzymes. We can come and cut using a certain enzymes and insect a different gene, and then we insect a different gene or a different strand of DNA there. When this thing here is going to unwind, when this thing is going to be during the protein synthesis or during the protein synthesis, the mRNA that is going to be produced here, you'll agree with me that it will be like this. And also having interpreted that new DNA that we have, and then also that gene will go like this. This mRNA will go and enter the ribosome, and after entering the ribosome, the strand is going to be interpreted. It means that the small amino acid, they will come and be joined here during the peptide bond. Up until now, another amino acid that will come and be joined there at that red line there. I'm not sure if you get this, but you have to understand this. You have to understand this is your thing to understand. When we talk about genetics and when we discuss this part, this is your things to understand, and you need to explain this by head. So you don't have to cram this. When you cram this, you'll have problems like what you'll be having now if you have a problem. So here's the thing. And then now we know that this can happen come mutation when it's happening natural, or it's being happening this change is being happening, what do we call, without us interfering. Do you understand? This mutation is happening itself, right? The change is happening there itself. There's not an alternate. Maybe the X-ray, we can do that intentionally, but this is happening unintentionally. We didn't plan for this mutation to happen, but since this mutation it's happening, it means that we have some things that they can go and cut that DNA and insert a new nucleotide. So we can also utilize those information, or we can utilize those enzymes to go to a certain gene now, to go to a certain gene, or to go to a certain protein DNA and cut it and insert a new nucleotides of our choice. And that nucleotide, maybe 3 of them, they are going to change that structure of that protein there to be something else. And then you know that these peptides of protein, they are going to form, these peptides of amino acids, they are going to form a protein. And then in this protein, now this protein will be having a red, having a something that is red there. So this protein is going to change its function, right? We are going to change this protein function to something that will work best or work less. For an example, insulin, right? Insulin, you did this insulin grade 10, grade 11, right? So this is what we are saying. We are utilizing that information that we got from mutation, that this mutation, these changes can happen. So now we are going to go there, intentionally, so remove a certain part of gene and insert the one that we like. And then they said you should learn the genetic engineering, right? And then here in this, they says stem cell research. You learn what is a stem cell research, and then genetically modified organisms. After genetically modifying these genes, right? The organism become genetically modified. You learn this, right? And then biotechnology. What is biotechnology? And then biotechnology is the chapter, and then genetic engineer. Biotechnology, it's like a chapter, and the genetic is a part. Genetic is a part of biotechnology. I'm not sure. Okay, we'll learn it when we go further. And then cloning, and then they said mention the mitochondrial DNA, and then paternity testing. So with our plan, you know, we go by the plan: Monday, Tuesday, Wednesday, Thursday, Friday, right? So this will be our plan. Monday, we are going to do genetic engineering, right? We are going to do genetic engineering, and then with biotechnology, and then Tuesday, we are going to do stem cell. You'll see why. We are going to do stem cells together with cloning, because this now we are dealing with the cells that they have been here differentiated, like both the blastocytes or other cells that are found in a bone marrow. Excuse. And then Wednesday, we are going to deal with questions. We are going to deal with questions either utilizing this information or adding this information to this. And then there's also, you also have to do the practical so we'll do the practical questions here, because these questions, they mostly incorporate them with practical. So you're going to do the practicals, the experiments, maybe like cloning. You go and watch a video where they are talking about the cloning, and then from there you have to bring the hypothesis, you have to bring the why, how can we make this experiment validity to be valid, or how can we make this experiment to be reliable, right? Then you come up with hypothesis, you come up with conclusion, you come up with the method of how do we do cloning, you understand, with a base knowledge. So before you can go and do practical, you have to have a base knowledge of what is happening, right? So when I here in this class, you learn this and then you learn this, and then Wednesday, that's where we can go and do those practicals, maybe like a cloning practical, and I will make sure how we'll do this. Maybe we'll come with pictures and everything, and then from there we did after Thursday we did Thursday we'll do mitochondrial links, and then because since this is the final week under this topic, we'll also do a small revision, maybe 30 minutes, and then Friday we are going to write a after question. We're going to, then where you will have the homework to go and do the last activity on our book, right? And then Friday we come and do the revision, and we do the answers to that questions, right? So I hope you understand our weekly plan. Monday, Tuesday, Wednesday, Thursday, Friday, right? And then I'm saying that Monday we are going to do a genetic engineering together with biotechnology, because biotechnology is the umbrella, or is the we can say we have a bucket, and in this bucket we have inside we have genetic engineering. Genetic engineering is a type of biotechnology. What is biotechnology? Is the technology of life, right? Yeah, I will talk about it when I go to the textbook. I just want you to understand this. And then Tuesday we'll come and do the stem cell, and the cloning, and then Wednesday we come and do the practical and the questions, and then Thursday we come and do the mitochondrial DNA and revision, and then Friday we come and do correction of the activity that we are going to write at the end of our textbook. You'll see it. So from there we can move on, guys, to our, this is what I was talking about. Now we can now incorporate the whatever I was talking about to our examination guideline, right? Examination guideline is said we have to do genetic engineering, paternity testing, although we did this paternity testing previously. And then under paternity testing, they just say the use of the following in paternity testing. How can we use blood to test for paternity? By this time, you know, we did this using the blood grouping, right? Where we did what do we call the A and the B, and then if like the father is like this, crossed with a mother with also she's A and B, and then this one they have a baby, and the baby she's a blood group O. So paternity testing, we are testing if these two parents, they are the true parents to this offspring, right? So we learn this. You'll agree with me that we learn this, and then after that you learn DNA profiles, right? The DNA profiles, it's when you do when we have those bars that you'll be given. Mostly they will give you this bars, and then they will say here's the suspect, and then here's the victim sample, and then the DNA was collected and was studied, and then we found the bars here. And then since this bar here of chromosome, this bar here of DNA is matching with the same bar here in a suspect in the victim saliva, or the victim sample, it means that maybe like during rape or what, right? So it can be the paternity testing, or it can be with these DNA profiles. You understand that it can also used for forensic, right? To find if the person, if this person, she's saying she's being raped, and then we have different suspects, then we can use this DNA profile to get to choose, or to get to pinpoint this suspect as the rightful person who's guilty, right? And then another thing is when someone is dead, we can go and collect like during this what do we call, during this accident that happened recently during Good Friday, may their soul rest in peace. Then how can we identify those individuals? how can we identify this person or these ashes that are here, maybe like an ash or a body, a burnt body, after the burnt body we have some cells, right? We go and collect the cells. After collecting those cells, we now bring the families or the relatives of those people. It means that we are going to have maybe if the bus was maybe 43, hence sometimes you just go there and then they don't prove it, right? Because here's the thing: if we have 43 individuals inside that bus and all these people died, right, and then we want to determine who those relatives based on the small maybe like particles that we find, remnants of them, right? Maybe a hand was found somewhere, then we want to bring back that hand to the rightful family, right? So what we are going to do, you learn this in DNA profiles. What we are going to do, we are going to collect the DNA of these 42 relatives, right? Relatives of these 42 individuals who are inside the house, who are inside that bus, right? So what is going to happen is that we are going to cut their DNAs and then add them into the jelly or that thing. You don't have to know what is happening, you just have to know the process of this is what is happening, like how I'm explaining to you, but it's a vast process of thing, right? Then after that we have to cut the DNA, put it here, and then each person will have his or her specimen. It means that we will need 43. If you are saying that your sister was there, it means you as a brother, you can go, or your parent can go there, right, and add that one sample of that person. So we want to find your relative here, right? So we are going to add that DNA here and we will have bars, right? So it means that out of those 43 specimen that were given, we are going to take each specimen and compare it with the DNA the DNA that we found on the hand of someone who was involved in that accident, right? So when we check these two, we then now check, this DNA band here, it at the same location as this DNA band here, because there's some variation and only 2% of DNA it's varied. That thing you don't have to know. This is what the basic information that you'll have to know. And then you also find that they also have accumulated here and they also accumulated there. We can now conclude that this hand that we got here, it belongs to Mang Mang Mang, and this Mang 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 belongs to this family Mang Mang and Mang Mang. So you see, it's a complex information, it's a complex process, but you also have to learn and you also have to know it. Or in DNA profile, we are using it as a forensic to identify the dead people, or to identify, do you understand? You can identify anything. You can identify, maybe like you just wake up today and then you find a cell, you find a tissue. You find a tissue of someone, or you find a tissue, a flesh, right? And then you go and identify the part of that flesh, where is that flesh coming from. Then you can go and then instruct the DNA and then go to the suspect animals. Maybe like you will say this flesh maybe but coming from a cow, a pig, a chicken and what, right? And then you put those DNA aside and then you did the DNA sample to the specific flesh that you have, and then after getting those bands, you compare, right? So we can also use that process as a paternity test. It's your father here, it's your mother here, only two things. Then we get your bands from your father, we get your bands from your mother, and then we have the DNA bands from you here, and then we compare. So if this is your father, it means some of the bands here, they will be aligned with the father, and then the mother, they will be aligned with the mother. So you get it, that. So we are not going to talk much about this. We are now going to talk about a genetic engineering, where we are going to divide this genetic engineering into 2 days. Today we are going to do a genetic engineering here, but you see, genetic engineering is an aspect of biotechnology, and includes, you understand, this is an aspect, this is another type of biotechnology. So what is happening here? It's a biotechnology. This is the technology of life. You understand? With biotechnology is the technology of life. That's where we are incorporating science and we are incorporating engineering into life. Bio means life, right? You understand? Bio means life. So we are incorporating those things into life. So we are bringing science and then we are bringing technology. Do you understand? Elon Musk, what they are doing there, those expensive those expensive laboratory activities that they are doing, trying to find specific body enzymes, the plants that they, you understand, that's what they are doing. We are incorporating technology and life, right? So this is what we are doing here. And then the genetic engineering, it's a type of biotechnology. In this biotechnology, we can do a part that called genetic engineering. We can also do something called fermentation, right? When we do you know that the beer, it coming from the yeast, you know, during COVID-19, we were doing we were surviving punch, pineapple punch, and then we were doing the enzymes, and then enzymes, they digest the juices and they produce alcohol. That was a biotechnology. We were using this technology to manipulate life or to better our, here's the definition here. Here's the definition, because this is a knowledge base, right? This is a knowledge base lesson. So I'm going to have a discussion. This will be a discussion with you, right? I bet the end of the day you should be able to also share this information with someone else. If you should be able to share this information with someone else, and you can be able to explain this thing to someone else, it means that when you understand what is happening in this concept. So right, I don't want biotechnology, but what is biotechnology in our examination guideline, right? If they ask you what is biotechnology and when you bring it like this, you are going to get the full marks. What is biotechnology? It can be manipulated, but this biotechnology is the manipulation of biological processes to satisfy a human needs, right? The manipulation of biological processes. What are these biological processes? Biology. Bio means life. Logical. What is happening kabuphilo? It means this process, they are involving life in it. For an example, the process of converting a juice here, a pineapple juice, into alcohol, it involves the living organisms. It involves something called a yeast, right? So when we manipulate those processes, when we manipulate whatever is happening there to satisfy our needs, we are now doing what is called biotechnology. Do you understand? What is biotechnology? In a simple man, biotechnology is the study, or is the a study of life using technology. Yeah. Bio means life, technology means the logi means the study of what? Study of technology. The technology means the incorporation of science and biology and engineering to better our life, right? So we use technology, like we use computers, we use technology is everything, right? Everything is technology. It was invented through technology. Everything that betters our life, or everything that makes our life to be easy, is technology, right? So we are incorporating this technology to the life. So in other process, we are manipulating a biological process to satisfy human needs, to satisfy us. For an example, we can use we can manipulate. Okay, we use a process here, insulin, right? We can manipulate a gene. We can manipulate a bacteria. We can manipulate this bacteria to produce insulin, right? This is the biotechnology. We are manipulating it. We are adding some set of genes into that bacteria so that bacteria will produce insulin, right? We are cultivating, or we are using a certain type of a yeast to ferment. You understand? We are using biological processes. Just like enough in this biotechnology, people now, yes, they will say in other books, is that people now, they can however change the gene. They can change the DNA structure of an organism. While they are changing this DNA structure, we call it a genetic engineering. It's an engineering that is happening at a genetic level. Mr. Paper. This is an engineering that is happening at a genetic level. We have Elon Musk, who are doing the spaceship, right? The Teslas and what, the electronic cars and what. That's an engineering that is happening somewhere. And then when you come to this now, you are dealing with an engineering that is happening at the genetic level. We are now manipulating cows to produce more milk. You are now manipulating a umbila to produce more umbila. We are now producing a now we are producing a genetically modified organisms. This organism, the organism that I'm talking about, when we produce maybe a cow that will produce now more milk, right? Manipulating these bacteria to produce insulin, producing huge mangoes, huge avocados, huge what, right? Doing this engineering at a genetic level. So you study this genetic modification organism today, and then they say the brief outline of the process. We just have to brief outline the process of with this genetically modification. We are going to use example of insulin. We are just going to brief that process of insulin, and then we do the the benefits of genetic modification. So tomorrow we'll do a stem cells research and a cloning. So from that, let's move on, guys. I hope you are now ready for our 30 minutes, my 30 minutes presentation of biotechnology, right? With our textbook, this is our beautiful textbook. We said biotechnology. Let me use people. Let me use people. So okay, 30 minutes, we are done with 30 minutes. Now we can go to our presentation using our textbook, right? And then they are saying biotechnology, but for centuries humans have used artificial selection to breed the best food crops, farm animals and pets, right? Think of all the different varieties of dogs that exist today. So here's the thing, back then they're saying that for centuries, using even people that were surviving long back there, they were using this biotechnology. Right, it started with biotechnology. Biotechnology is the technology of life. This is the technology of life, right? Technology of life, or a technology, applying technology to the life processes, right? So when even to take a certain dog and breed it with a certain dog to produce a pit bull, or to produce a breed, or one dog that has a long legs and a tall body like a horn, those tall dogs, and the pit bull here, that new animal. We've been doing this, or humans, they've been doing this for a long time, right? Artificial selection. When we talk about artificial selection, this is a selection that is not happening naturally, where now you go and take a certain animal and breed it or mate it with a certain animal to have a specific and offspring, right? But with modern science, however, technology, science and technology, however, humans can manipulate the actual DNA of organism now. We used to say, hi, back there we were just doing this thing of if we can take a donkey and mate it or make it to sleep with a horse, we are going to have something called a mule. If you know this animal, a mule is it's a hybrid between a donkey and a what we call donkey and a horse, right? So they started doing this long back there. But now we have science and technology. And with this science and technology, we can now go straight to the cell itself and manipulate that gene. We can now go straight to the chromosome and manipulate that gene. We can now extract, or we can now come to the horse and remove a certain protein or a certain DNA and take this DNA and implant it to a human being, and that human being now becomes muscular. Do you understand? Now here we are creating new organisms. Here we are creating new species in this biotechnology. But you can see that they only give you a brief explanation of what is biotechnology. It means that they don't want you to dwell much on what is biotechnology. You just understand that in technology, we are using technology to better life or to certify ourselves. But biotechnology, in the next paragraph, but biotechnology is the use of organism. Here we are using organism, right? They're saying in biotechnology, we are using organisms such as bacteria or a biological process, right? To improve the quality of human life. As for example, doing DNA profiling, right? Doing DNA profiling this process of cutting the DNA and edit inside here is the example of biotechnology. During genetic engineering, when we can go to a gene or DNA structure level and remove some nucleotides and add some nucleotides, we are doing a genetic engineering. We are engineering into a genetic. It's also type of biotechnology. They said stem cell technology. Stem cell technology, this one will do it tomorrow. This one will do it tomorrow, but we'll be talking about the cells that they haven't differentiated, right? It means that we are talking about the blastocytes, morula, zygote there, right? So we talk about those cells, right? And then after that, we talk about cloning. That if we have a cell like this, you know that it's a diploid cell. It have a 46 chromosomes. So if we can come and remove this chromosome and add another chromosome from a specific cell, it means that we are going to have someone that is identical to this person. That's now we're talking about cloning. You did this in grade 10, but we'll also talk about it tomorrow, right? So but DNA proofing was dealt with chapter 1. It is a form of biotechnology also used for paternity testing, right? But the identification of individuals, like what I was talking about, those who are involved in a car accident, those who are involved in a bus accident, right? May their soul rest in peace. And then for many other purposes, DNA profiling, right? We can use it for many other processes. Someone during a rape to find a victim, you understand? But also here, I will concentrate at the paternity testing and identification of individuals, right? So when now you can have a sex, you can rape someone, and then they go to that person and they instruct the semen that you released inside that person's vagina, and then they do a DNA profiling. They will catch you, or you are the person who raped this person, right? So they take your DNA, you know this, they take your DNA, and then you are a suspect, Anir. You take the DNA and then they go and match with the DNA that is on the sample. And if we find that some bands here, they are the same, you are the person who raped that person. And then we are going to tswar your saver number. We are san sayan, your san what more detail. Primrose what. Right. Let's move on, guys. I'm tired. So we talked about a biotechnology here. What did I miss? Nothing. Now we are going to talk about humans can manipulate an actual DNA, right? So when we are manipulating this actual DNA, we are doing a process called genetic engineering, a genetic that is taking place at a genetically level, right? And it's also part of biotechnology. While in this biotechnology, we are using some organism. We are using another organism to better our life. In biotechnology, you can use a plant, right? You can use an animal, or you can use a microbes. These are microorganisms, right? So like bacteria here, you can engineer the DNA of all these animals. So we are using an organism such as bacteria, or even animals or plants, or a biological process. When we talk about the biological process, maybe on the production of a certain something, right? Then while you are producing that certain something, we came to your DNA, and then we manipulate it. After manipulating that DNA, we make use of that biological process. And then like insulin, you learn this when we do insulin today. And then all that, we are doing it to improve the quality of human life. Whatever that we are doing here, we are doing it to improve the quality of human life. So here you can use a career in this topic here, where you can become an engineer, a genetic engineer, guys. I think we can also apply, most of us, guys. We can be a genetic engineer, guys. Being an engineer dealing with genetics. Imagine that today they will tell you, hey, the disadvantage of this is because it's expensive. The disadvantage of this because it's expensive. Hey, what. But we can start our field, guys. Right? We can start our field where we can just use cows. We can use a chicken, right? And then maybe actually you know that we as we become one team, we will need others to be agricultural side. So those who will be in agricultural side, those who will be plowing some certain animals, certain plants, sorry to say, those who will be farming certain animals like cattle, or the sheep or the pigs, right? So they can bring their pigs to our labs, and then Rina as the genetic engineers, we take a cell from the pig, and then we add it with another cell of the pig, and then we produce pigs that are huge, maybe advantage, that are huge and that have fats, maybe for the bacon side and what all that we are using it to qualify or to qualitate or to increase the quality of human life, right? I hope you get that. And we use other organism. Okay, so now let's go to this type of biotechnology, which is called genetic engineering, right? So now we have learned what is this biotechnology, and this biotechnology, we have a type of biotechnology called a genetic engineering, engineering that is taking place at a genetic level. It means that we are going to cut a gene now, open a gene, add another nucleotide, perform a mutation. Yeah, we can perform the mutation on a certain gene, right? We can change the sequence of nitrogenous bases, right? So okay, but genetic engineering is used, this is the process, right? This is the process that is used to alter the genome of a living cell for medical, industrial, or agricultural purposes. So here's the thing, this genetic engineering, this is the process that we use to alter or to change or to manipulate. We are changing this, or we are manipulating this, manipulating what? We are manipulating a genome. What is a genome? What is a genome? A genome is an entire set of the DNA of a human being. Your cell, let's say, like, let's take it like this. Let's do like this. Your cell inally DNA, and we have the nuclear DNA, and we have a mitochondrial DNA. The entire set of DNA inside this cell, it's called a genome, right? So even on a bacteria, a bacteria is something like this, right? It's a unicellular organism. It's a one cell organism. So in this organism, we have two type of DNA. We have a DNA here, and we also have a certain DNA that we are going to learn about it in this chapter called plasmid, right? Plasmid. So all this DNA, including this is called something called nucleoids. Let me write it. Nucleoids, right? These are the DNA in nucleus, and then we have a DNA outside of the nucleus called a plasmid. This is an outside DNA. We have two types of DNA in bacteria, right? So we are using, or we are altering, right? We are altering, or we are coming and manipulating this plasmid here. It means that we are going to cut this plasmid, and after cutting this plasmid, we are going to join it with another DNA, and then we are going to force this bacteria to manipulate something that it's manipulating, and then manipulating our DNA there, right? We can use it in cells, living cells, for medication. We can use this process to produce insulin. We also have another many processes that you can use this to produce. We can also use to produce hormones, like those that are now being transgending. You take hormone pills, right? You take pills for testosterone, you take pills for what, and then we find girls becoming babali the beard, right? So you can use we can use this genetic engineering for many purposes, but you know that our education is limited to this information, this kind of information. So here the example that we are going to use, we'll use insulin as an example, right? And then we can use it for industrial. Also insulin we can also use for industrial, for the production of insulin. We produce more insulin, we can produce vaccines, and then after that we can now ship those insulin of ours to different countries, right? Or for agricultural purposes, agricultural processes, also using a bacteria. We have bacteria called agrobacterium, right? We call them agrobacterium. Can I write it down? Agrobacteria. I'm not sure if I'm wrong. You will comment the agrobacterium. This bacteria, they go and inside their gene, they cut a certain gene and replace and add another gene of their likings. Or we will be now inside the lab. We'll be doing this inside the lab as a team. So this is what we do. We add a certain gene inside and this agromium, and then we take this we take this gene now and add it to a certain plant, and then that plant is going to grow. We are going to manipulate the growth of that plant. Maybe for an example, okay, I'll come here down here and do an example. Let's take for an example, we have a fruit, right? This fruit here, it's long, and then this fruit, it's having a small juices inside, small fruit inside, right? And then we also have a one tree here that is producing big what, big fruits right inside this. We can, so these are the peas, right? Since we have been doing peas, so in this chapter we are saying that we can go to the DNA level, and we take the DNA from this plant here and combine it with a DNA from this plant here. We can say a gene and combine it with this gene here, or a DNA, a part of a DNA, right? And then the result that we are going to get here, to certify human needs, we are going to find a result of, now we are going to find a tall fruits that's having a huge fruits inside, right? This will be the new DNA, right? This will be the new characteristic. We are manipulating two characteristics like this, right? So when we are dealing with this, when we go to a gene level and now straight to a gene level and try to remove this gene here and add this gene here, maybe try to cut here and add this gene here, whatever we are doing in this process, it's called genetic engineering. We are engineering. We are building our own organisms now. We are building our own machines, and then yes, they will say that this thing, it's also varying. This thing is also they say this is ethical people, that they don't want the religious people, right? They will say, hey, you are making yourself God, right? You cannot tamper with organisms. Do you understand? You can't just go around and make a huge we take a, maybe for an example, we take DNA from elephant, and then we take DNA from the cow, and then we mix them, and at the end of the day we have a huge a cow that looks like an elephant, right? Hence we don't do this, because there are people that they don't approve that. They deem them, or they refer this as satanism, right? Or we are not sure if maybe someone is just using that information so that us Africans, or us Africans, who this information, or this information that you are getting here, it came here colonized already. They are just saying it so that we don't utilize this engineering side of thing. Do you understand? Imagine producing elephants, or we producing cow that is elephants. Do you understand? The huge cows that now we can feed all of Africans. We produce the huge corns, umbila. Actually, we live in pap. We eat pap. So we produce the huge umbila that only one corn thing can make a 1 kg of meal with this genetic engineering. So let's go and let's read and find out more about this genetic engineering. But the results of genetic engineer, these results in a genetic modified organism, GMO, or transgenic animal, right? Animal with DNA from more than one species. So I'm not sure here even the transgenders, they are referred to genetically modified organism. I'm not sure, right? But also what we are doing with those transgenders, you know that we are, you have a boy having the reproductive structure of a boy, but we are going to insect a hormones, that these hormones, they are going to activate his bum growth and his what we call the breast growth, right? So we are not sure if that person now is the result of genetically modified, and then we can refer to him as a genetically modified human. I'm not sure, right? We still have to learn that, or I still have to make a thorough investigation under that topic. Yeah, or a genetically modified organism, a transgender like this, if you are saying you are a human being, you have a reproductive of a human being, but you are masculine, you look like a man. Do you understand? Can you say that you are genetically modified? Although we cannot modify your reproductive system. A reproductive system, we can genetically modify it during what we call during fertilization, right? The first type of fertilization, if we can fertilize an ovum with a sperm that is housing an X chromosome, right? I'm not sure if you get that, but that's what I'm trying to explain. But after that, when these two fuse, when this zygote happen, there's no way that we will be able to alter this sex here, because the moment the X and X go and match, the fertilization is just one chromosome from the mother is matching with the one chromosome from the father, and so forth and so forth up until chromosome X match with chromosome X, and then we now say the person that is going to be produced here, she's a female. Or X and Y, we say the person who's going to be produced here, he's a male, right? So, but this, and then this genetically modified organism, you can find them behind your food. You see that in some food, they'll say this food does not contain genetically modified organisms, right? GMO. This is what they are talking about. They say GMOs not included, maybe on a meal or on rice, right? They are saying that because we can do this in plants also. We can do this in animals, like a beef. We can say this beef does not contain GMO or GMO free. We are saying that this beef was not produced by a genetically modified what? Cow. Right? If it's a rice, we are saying that this rice was not produced, it's an organic. Yeah, sorry, thank you. They say this thing is organic. It means that if it's a plant, it grow up naturally. Although today we can say something is organic while they are adding some pesticides, or they are adding some chemicals that they get absorbed inside the plant and then go to the DNA structure and alters the DNA structure or form a mutation, and that mutation we end up having a plant that maybe they can grow taller, but because they are growing taller naturally, and these seeds were not genetically modified, they end up referring this as a genetically modified organism. Hence, the process to grow this plant, it genetically modified that organism. And like the example, we will use chickens, right? We will go to the Woolworth tomorrow, during weekends, go to the Woolworth and check. We have chickens, and then in that chicken, they will say this chicken, this is a drum thick by the way, they will say this chicken is a GMO free, right? Genetically modified organism free. It means that this chicken here, it was raised up or it was obtained from an organism that was not genetically modified, right? But here's the thing, this chicken, they give them, or they feed them food, or they feed them some yeah, we can say food, right? The feeding thing of the chicken. We know this thing. This is what is happening every day. Somewhere, when you have a chicken farm, there's a special food that when you give those chicken increase in size, right? And then we know this thing. So this Woolworth thing, Woolworth, Pick n Pay, what I'm just using an example called Woolworth, right? Because they will say it's a high standard, it's a grade A chicken, right? So here's the thing, the food that they will feed this chicken, this food contains amino acid or contains hormones that when this chicken here, these hormones are being ingested to this chicken, it's going to cause this chicken to increase in size, right? The food that they are giving them, they contain amino acid, or they contain a nucleotides that they are going to alter the DNA inside this chicken, and this chicken will result in becoming big in a short period of time, right? So when this chicken becomes big, because they are saying this chicken the genetic structure of this chicken was not altered, right? Altered yet. It was not altered before this chicken was born or after this chicken was born. We can now classify this chicken as a GMO free, genetically modified free, right? It's what something that now I'm saying, we can say this, although the genetically structure of this chicken was not modified for this chicken to be like this, but the things that this chicken got fed, it went inside their genetically level and altered that genetic level and produce something, or produce new proteins that now forces that chicken to be big. Is that not a genetically modified organism now? Hence now you find that where now you go and eat this chicken, agree, this chicken is going to produce that protein, and that protein is going to be stored into the flesh of this chicken. And then when now you go and eat this protein, you digest that protein. After digesting that protein, you have those amino acid, and then you ingest those amino acid, and those amino acid, they go straight to your cells and produce a certain protein. And that protein now function whatever it was functioning inside this chicken, and it's going to manipulate your body, maybe to store more fats, or maybe to increase your body muscle, right? So you end up now becoming big, right? Or I'm revealing something here, right? Maybe or maybe not. Those who are following me, guys, please comment. This will be your first comment on this 43. Please comment, tell me what you think about this, right? Tell me what you think about this, guys. You go and eat the chicken, and the chicken contains the amino acid, that this amino acid, they are going to change. Let's take for example, your DNA is like this, and your DNA it's a T, right? So amino acid here, mRNA here is going to be uracil adenine, right? So you find that this uracil adenine, this it's going to match with a tRNA, that is going to be adenine uracil, right? So you find that this tRNA, it's holding a specific protein, right? A specific amino acid. But because now you have a different amino acid being introduced to your body from the chicken that you bought from those supermarkets, right? It means that when now that this amino acid, instead of bringing this amino acid, it's going to bring another set of amino acid to this protein that you are producing in your body. So that protein, that protein that you are producing in your body now, it now come with a new amino acid, right? And you know what's happened when this happened. It means that the new protein will be produced that will function something completely different, or partially to what it used to function. So the chicken here, they can say this chicken is genetically modified organism free, but it contains, or its gene, it was altered by the amino acid that it was fed with, right? I hope you understand that. So let's push and finish this, right? Let's push and finish this chapter here, and then this one we'll talk about it on the comment section, right? We'll talk about it on the comment section, guys. Then okay, let's move on. And then they said genetically modified organisms are used to this organism that we modified, right? Let's say maybe like we have a bacteria, and that bacteria will go to its DNA and change it intentionally. We can use now to breed more productive crops or animals, so that more food can be made. So we can have a genetically modified dog, a genetically modified cow, that we can take this cow and mate it with another cow, so that we produce more cows, that they produce more milk, right? Here in animal, or even in crops, increase yield. They say increase yield, and then we go to the crops, we go to our mealie meal, our umbila mealie meal, and then we can now use the genetically modified organisms to produce more mealie meal, right? And then another bullet, they say to produce drugs or hormones, right? Insulin is an example, which have fewer side effects and they're cheaper. So we talked about this. And then to infect cells to cure diseases, gene therapy, such as brain tumors and fibrosis. Here what we are doing is that we are going to the cell, and then we insect it with a certain gene, so that that gene will go and be interpreted into something that now it will cure that diseases, right? So Barry, one process used to produce a genetically modified organism is called a recombinant DNA technology. A recombinant DNA technology, like what the word is saying, a recombinant DNA is a recombined DNA, right? A recombined DNA. So this is the process that we use. A recombinant DNA technology is the technology where we cut a certain DNA. You will see it, we'll do it on the next slides. But it can be used to manufacture a human insulin using E. coli. a bacteria, the process can be summarized as follows, right? So we are using a bacteria called E. coli. So this is the process that this thing undergoes. They will ask you maybe to explain or to give a brief explanation of this activity here, right? So today we'll end up here. Our time is up, and then tomorrow we'll come and do the benefits, the what do we call the come on, the advantage and disadvantage of this recombinant DNA. But for now, I've said some of them, and I've said the benefit of them. The benefit advantage is that we will have more crops, and then another advantage is that we will have those products that are being used. They have more shelf life, like they don't spoil fast. And then another advantage is that we can have more food, so we can have a food security. And then another thing is that we can cure diseases. Those are the advantages. We can cure diseases, or we can curb the spread of some diseases. And a disadvantage, this process is expensive. So someone might come and say, hey, the reason of this thing being expensive is no longer validated, or it's no longer valid. Hey guys, this thing is expensive. We're not to have machines to come and cut, to have enzymes to come and cut this DNA and insert a new DNA. It's not that much cheap. Maybe you can take a cow and force it to have sex with buffalo, and we produce some cows that maybe like they produce faster, like they have 2 babies per year, right? That's the least that we can do as a biotechnology or as an engineering. But another disadvantage is that when this thing is happening, as you can see in this picture here, we have a process that is happening. When this process is happening, we don't know, or we are not sure if a certain, because due to uncertainty of a specific gene. So we might take a specific gene thinking that the gene that we are taking is a correct gene, and then we find that we incorporate a wrong gene, or something happened there during that process, and that thing, so this thing becomes what we call, it becomes risky, right? It becomes risky. What if we produce now animals that those protein are killing us, same as chicken, right? So with chicken, we don't know if we don't have a long run, because Woolworth, what I know, we need. Now more people are affording Woolworth. Back there, most people they were not even what do we call, being affording the Woolworth. So okay, it's fine. We can do advantages and disadvantage of this genetic engineering, or this genetic modified organism tomorrow. So today, let's do this practical. This will practical that you are going to do, or this is the experiment. The experiment called recombinant DNA technology, right? Recombinant. Say recombinant. Recombinant DNA technology. So we are using technology, and then we are using technology to combine two different DNA to produce a specific thing that will benefit us. So this is the technology. We have some things, you understand, you still have the eye tubes, you still have the tubes to grow the cells that you are being produced, you still have the incubator to incubate this. So there's a lot of this. So the technology is very much vast here. So right, so in a nutshell, what is happening? This is a nutshell. There's a lot what is happening. There's like the thing of enzymes and what we don't have to know about them, right? So here's what is happening. We have a cell, right? We have a bacteria, right? This is the bacteria. And then one thing that you'll have, this is the bacteria here. This is the bacteria. And one thing that you have to know here, we just have to know that this is the bacteria, and this one is something called the plasmid, and then here it's called the bacterial chromosome, right? Or a nucleoid. Why not just know that we have a bacterial chromosome, and we also have a plasmid. These, all of them, they are DNA, right? The chromosome, chromosome is something like this, but it's also made up of DNA. And then we also have this DNA that is not called to anything, and it's a cycle form, right? So the beginning of this thing is that we'll find a bacteria. Our bacteria will be like this, and then we will have these plasmids here. right? And then we will have the one chromosome, the bacterial chromosome here, and then this bacteria here, it will have something called a flagella. It's a tail, a flagellum. Then you'll see this a bacteria because of something like this, and it have the ring thing, and this ring thing we call them the plasmid, right? The plasmid, it's a type of a DNA, and it also is a chromosome. It's a type of a DNA. So these bacteria, they have two types of chromosomes, right? They have two types of DNA. So their genome comprise of a plasmid, right? You'll agree with me, and a chromosome, this bacterial chromosome, right? So our genome, as a eukaryotes, it will contain DNA inside the nucleotides, inside the nucleus, and then the DNA inside the mitochondria, right? So we come and manipulate this plasmid here of this bacteria. This plasmid here, you can see that we cut, this can be the step 1 or this can be the step 1, both of them, right? You can come here, no, this will be the step 1. Why? Because before we can come and cut our thing here, we have to go to a patient and isolate a gene, or extract, right? They will say extract or isolate or take out a gene to make insulin from a healthy pancreas cells, right? So you understand, or you're going to learn about this pancreas, or you did this last year, right? You did this in grade 11. You are also going to learn about this during an endocrine, the response to an environment, right? So in these pancreatic cells, we have cells that produce insulin, right? There's a cell that produced insulin. So here's the thing, inside that cell, you'll say the nucleus, and then we'll say the mitochondria. Inside these cells, we have a DNA, right? We have a DNA, and then in somewhere in this DNA, we have a gene. And when this gene got interpreted by mRNA and the message is sent to the ribosome, we produce the proteins, and those proteins, we say they are the insulin, right? So we come to this cell inside the pancreas cell. We come to these pancreas cells and we open it, and then we use some enzymes, right? Cut out using enzymes. What the names of the enzyme, they say we don't have to know. We not just know that we come here and we use a certain enzyme to cut this gene here, right? After cutting this gene, we can now have it. It's as a double-stranded, right? A double strand. This is a gene. This will be the gene. And then after cutting this gene, this will be the gene, right? After cutting this gene, we take this gene using the same enzyme, using some enzyme, right? I agree we use enzyme to cut this off. We are going to use some enzymes to add this into this plasmid of ours, this cycle of ours. So it means that in this plasmid was a full cycle, and we use some certain enzyme to come and cut this part there, right? But cut open the bacterial DNA, which is plasmid. You can say cut the bacterial DNA, or you can say plasmid, or they can give this in a single word question, right? A DNA, a bacterial DNA that it's used during genetic engineering. Well, now you should know that it's a plasmid, right? So when you are explaining this, you can just say cut open bacterial DNA. Which bacterial DNA are you talking about? You can now say it's a plasmid. Using another restriction enzymes. They will say these are the restriction enzymes. Restriction enzymes, what they are going to do, it means that they are going to restrict this joining of this what do we call? Come on. This piece here. I agree we understand that each and the DNA, there's a difference between DNA and RNA. And in our DNA, we know that adenine match with thymine, guanine match with cytosine. In all type of DNA, where there's a bacterial DNA, is what chromosomal DNA, chloroplast DNA, and adenine is matching with a T, G with a C. So we come and cut here, and then maybe like the next year was going to be G and C. So we have enzymes that can come and cut this part here, and also that can come and part here. cut this part here, right? And then when they cut this part here, this plasmid or this ring becomes open, right? And then we go to a human being and we will want or we will need the enzymes that when we cut here, we will have maybe here we'll have A that was merged with T, and then this side we will have G that was merged with C, right? So this thing here will come and, do you understand, fill this this portion that we removed here, right? So what we do, we come and cut this DNA here. We have the piece here, and when we have this piece here, we come and cut this part here. When we cut this, we can just remove two group, right? GC and AT, and then we bring the other piece here that will merge these two, right? And then when we merge these two, we are going to have something like this, right? And then number 3, insert the chosen gene into the plasmid and attach each end using enzymes. So we are using enzyme to cut this DNA here. We are using enzyme to open this plasmid here. We are also using enzyme to glue this together. So we have an enzyme to glue, and we have an enzyme to cut, and we also have the enzyme to cut. And then what they are cutting here, they are restricting enzyme, because after cutting here, they also wait here so that these and those parts that we removed, they should not come and also join there, right? And then when we have something like this, now we have a full ring of a DNA, double strand, right? We have a full ring like this with T adenine, guanine, cytosine, guanine, cytosine, what, adenine, you understand? A and T and G and C, right? So you understand this. From there, we take that DNA, we take this DNA and insect it. We remove it. Then now after having it, we return it back into the bacteria. As you have seen, our bacteria, it has more of plasmid, right? So we just insect another plasmid here, and then this will be our plasmid inside this bacteria. We will have a plasmid like this now, with another half from a human being and another half from a bacteria. So what this bacteria is going to do, this bacteria is going to function as normal, and we know that functioning as normal, the bacteria, they undergo binary fission. Do you understand? It means that one bacteria grow up twice its size and then divide into 2. So it means that this bacteria is going to, this half is going to divide like this, and we are going to have one half there, one half there. The plasmid here will undergo DNA replication, while after DNA replication, we will have exact copy of these 2 DNA here, and then hence now we find one DNA this side, right? So that is during the DNA replication. The infected or the insected bacterium here, but the bacterium is now genetically modified and will produce the desired protein. How? So here's the thing, this bacteria here actually started as 1, then it's going to divide, and then after dividing, it will form these 2 daughter cells. This daughter cell, they are also going to divide, binary fission, right? You know binary fission is just a division of a bacteria where one bacteria divide into 2, right? So even there, we are going to have another bacteria, same composition like this, having a 2 color, a plasmid and a thing, right? So when that bacteria is moving, and that bacteria now is doing its daily thing, right, it means that that DNA there is going to undergo a process called protein synthesis, right? You know, protein synthesis, and you know, with protein synthesis, the mRNA will come and take the DNA strand and remove it and take it to the ribosome. Inside the ribosome there, it's going to be interpreted, and while it's being interpreted, we will now include that gene that we inserted, right? There's something will be like this. Now you'll agree with me that something like this will happen, right? So these DNA, these peptides here that are being produced, they are going now to be linked with this protein here. This, do you understand? So here's the thing. Because now we understand that this gene, right, this will be the gene, right? It means that during DNA replication, during mRNA producing, during protein synthesis, this gene here, this piece of gene is going to be interpreted as one mRNA. You get me? So we'll remove actually this gene, this DNA. You know that this all of this DNA does not code for protein, right? It means that we'll come to a part where we'll remove an mRNA, and this mRNA will just be having this information to produce insulin. And when we attach it to the ribosome, it's going to produce a protein. That protein is made up of amino acid. When those amino acid combine, they produce a protein called insulin, right? Insulin. So now these cells here will have insulins inside that's moving around here, and then we can now I'm not sure if this insulin they come out, maybe they get expelled from they got expelled from the bacteria, or what we are going to do is that we are going to open this bacteria, we're going to crush this bacteria and collect this insulin inside. So that's the information that I'm not sure if the entire bacteria, after producing an insulin and realizing this is an insulin, maybe it's being produced. These bacteria, they work wonders, right? You might find that it will cause something like this, and then we exclude that protein, and then after that, that protein is released. Anyway, this is happening inside a liquid thing, right? So these bacteria, they will be incubated inside a liquid thing, giving them an optimal temperature for them to reproduce and to replicate and to transcribe their DNA, right? So either way, if the insulin is being produced, the insulin can be collected from the bottom of the tube, or if it's not produced, it means that we'll need to crush this bacteria and we collect the the insulin inside. But number 5, the bacterium can now be cultured to form many more bacteria which can act as a factory, e.g. to produce insulin. So now with these bacteria of ours that they can now produce, we can now take other bacteria and place them and cultivate them, place them into different cells, and then we feed them with whatever they need for them to reproduce and produce more insulin. And then we have a company, and that company now it's registered and it can produce insulin, and you know how much we can make from this process. So it's not only insulin. So you'll understand from there, we'll do advantages and disadvantages of genetically modified. So you can go and study those since we have already elapsed our 1 hour. Guys, this is what we'll talk about in recombinant DNA, right? So it's not only insulin. We can come here and remove, we can go to a certain cell, maybe like hormones. I was talking about hormones, testosterone. We can go to the testis, the cell of Leydig, still they produce testosterone, or we can go to the cells that produce hormones, and then we remove a certain gene, and then that gene we insert it to a plasmid. After inserting it to the plasmid is formed, and then we take the new plasmid, a genetically modified plasmid or genetically modified DNA here. We take that genetically modified DNA, we insert it or we put it back inside the bacteria, and that bacteria now it becomes a genetically modified organism because it's having a genetically modified DNA. When that bacteria now is having that genetically modified DNA, we can now plant them and collect those proteins that they were encoded inside that gene. Right? You get me now? If like now we are producing testosterone, that gene now we are going to say remove the testosterone inside the bacteria, collect that testosterone, and then we sell that testosterone. So you see, you can have a business. We can have a business with this. So guys, that's it from me. Don't forget to subscribe, guys. I beg you, don't forget to subscribe. Don't forget to comment. Don't forget to like, guys. Don't forget to share this with other learners. Maybe like you cannot share the entire video, but you can share the WhatsApp link. You can share the group, the YouTube link to other students so that they can also join, and we have a huge class, guys. Let's increase our class. I'm also going to post this on our TikTok page and on our Facebook page, and I'm going to beg you guys to help me and reach out to other students, guys. So thank you for being here. Thank you for subscribing. That was it from me. We are done, guys. Today we spent a lot of time, but this is it, guys. Let me also go and do something else. But with biotechnology, a recap today. Let's recap what we did today. Today we also, it was a knowledge based, right? Knowledge based class. And then in this class we learned that we have biotechnology, and there's a difference between biogenetic engineering and biotechnology. Biotechnology is the use of organism and or bacterial process to improve the quality of life, while we are improving the quality of human life. We are using the geological biological processes, or we are using other organisms to produce certain chemicals, certain food, to understand. And then from there we talked about DNA profiling, and then we talked about genetic engineering, where we are now going to the genes itself and we manipulate those genes, right? And then the organisms that they contain those manipulated gene, we call them genetically modified organism, right? And then we also talked about the transgenders and what if they are genetically modified humans, right? From there we talked about how these genetically modified organism, we can use them, those bacteria that we were talking about, that we were cutting the plasmid and inserting those plasmid, we can use now to produce hormone. Here we can use to produce drugs or hormones such as insulin, which have a fewer side effects and they are cheaper. So you see, this thing is cheap. So whatever hormone that we can produce, guys. If maybe we are saying that we are now where now you want cows, you are doing cows, right? And you want cows that are huge in the shape, so that you have you can increase the yield of meat, right? Or the yield of milk, right? Even both in chickens, we can increase the yield, or we can increase the production of meat, or we can use the production of egg, right? Since the egg, now you say they were so much expensive, so we can go to the chickens and select those chickens that are producing eggs, and we take their gene, and then after taking it taking their genes, and then we insert it to other with another genes that maybe like those chickens, they are huge. You understand? We have a huge chicken and the chickens that produce more egg, and then now we end up having huge chickens that produce maybe chickens, right? Or we have chickens that produce huge eggs, large egg, and those that produce a small egg, right? So we can now take the gene from this the big egg and combine it with those that produce small eggs but they produce more eggs, so that we will take the gene of more eggs and add it with the gene of big eggs, and we end up having more big eggs, right? Those are also the advantages that we'll talk about tomorrow. That the advantage of this is that we can use this to increase the food production. We can use this to produce the medication. Maybe you can know 2 or 3 advantages and disadvantage. And the disadvantage is that this thing is expensive. And another thing they say, another thing here, disadvantage, this DNA here, we have a unwanted gene being transferred from one generation or to other individuals, right? So for example, when now you have these eggs, so this egg, inside them they will have that gene, right? They will have that gene. So when as a human being, you will come and eat this egg, and that gene is going to be printed in your DNA, and now we end up having unwanted genes inside our bodies. Hence now we find a huge grade 12 students. Now you grade 12 students, you are huge, right? So all of them, it's happening here at the genetically level. So that's the disadvantage. Another disadvantage, this process, they are expensive. And another disadvantage is that we end up having unwanted gene, and then we end up not having diversity, right? We end up having same animals or same kind of animals. If we are talking about for an example, pigs, we end up having one type of pigs, Do you understand? Like dogs who end up having one type of dog, who's sportic or high, there are many because they inbreeded with one another, and that gene was passed on from different with different dogs inside into that village, and we end up having most sporty bunch. So you get me? So we can use this genetically modified organism to breed. To breed is to take one animal and mate it with another animal. More for more production of crops. So we can take a plant here. That plant produces a small meal, but many. Yeah, let's come and do example. That mealy produce that mealy tree produced more mealy but small, right? But we also have another tree here, and that tree produce less but huge mealy, right? So we can take the DNA from this, and we take DNA from this and combine it, and we end up having a tall millers that they are huge, right? Depending on the gene that we take here, we take the huge production, and then here we take the gene of many production. We combine these, they have become many huge product, right? Even on animals, we can use this to have more animals that are big, or that are that produced more food, more milk, and that, right? This is the information that you have to know. And then what are the to infect cells to cure diseases, right? We did this gene therapy such as brain tumor and cystic. When you are banned, right, we have here, we'll be talking about stem cells. Here we'll be talking about the stem cell. We can take stem cells from where we find them. We can learn about the sources of stem cells, and then we insert them into a certain body part or body tissue, and those cells, they are going to cure those body tissues, right? So that was it. You learn that, and then you learn the process of DNA recombinant. How do we recombine gene? How do we use one DNA, combine it with another DNA to produce a desired protein, right? This was the process. You learning that we come to the certain specific gene of specific cells that we want. Here we went to the pancreas, because the pancreas, it contain a cell of islet, right? Cell of islets that produce insulin. So we'll come to the cell of islets, or we go to the leg cell of Leydig, something like this, if I'm not mistaken, inside the testosterone, inside the testis, and then we remove this Leydig, and then we take the DNA, we combine it with the DNA plasmid bacteria, and then we produce we have the entire DNA. We take the entire DNA and insert it inside the bacteria. After inserting inside the bacteria will now think that the bacteria will have to interpret all this DNA, and while the bacteria is doing that, it's also interpreting a genome, that gene is going to produce an mRNA. When that mRNA goes inside the ribosome, it's going to produce proteins that they will be testosterone, and then we collect that testosterone, and then we can produce factories. We can now have factories that will sell this specific protein. So you see, we can also have business here. We can have you working as this when you grow up. So that's it, guys, for me. Thank you. Today it was the longest class, and it's Monday. Guys, let's recruit others, right? So thank you, guys. That was it from me, Mr. Mpepu. Then tomorrow we'll do briefly what do we call the benefits, the advantage and disadvantage, and then we'll go to the stem cells. So thank you, guys. That was it from me. Let's meet tomorrow, same time, same place. Peace.